Hey everybody, this is Miss Peachy from Wisconsin Connections Academy, Biology A. And today we are doing, or I should say I am going to be doing a little tutorial for Unit 7, Lesson 4, Mitosis in Complex Organisms. So make sure you are clicking on the link. It's actually going to be in the lesson. I believe the link's above the video now since they moved things around a little bit this year. And it is for the study guide that goes along with this lesson. So our keywords today are differentiated cell, differentiation, extra embryonic cells, gene expression, um, multipotent cells or multipotent cells, I don't know how you pronounce that, pluripotent cells, and senescent cells, and totipotent cells. Wow, mouthfuls for sure. So we'll talk about what that actually means here. Well, that's not a very attractive picture of this zebra. So anyways, we're going to kind of go through the lesson and just kind of go through some of the main points here. So we are looking at eukaryotic cells. Organisms that contain chromosomes within the nuclei of their cells are eukaryotic. You should know that already. Organisms that don't do that are called prokaryotes, right? So what makes us special, not just us, but other eukaryotic organisms, is that we have our genetic material contained inside of nuclei. So mitosis can only occur in cells that have nuclei. So it can only occur in eukaryotic cells. And another important point is that when mitosis happens, it makes an exact copy of the cell. There's no genetic diversity at all. It's like a photocopy or a Xerox. It is a clone. So genetically exactly the same chromosomes, same copies of those chromosomes. And that's important because a lot of times mitosis is taking place because you're growing or a cell gets damaged and needs to be replaced. And so you want to make sure that that cell is exactly the same as it was before. Mitosis is just part of the cell cycle, and the cell cycle is made up of multiple different parts. A big chunk of the cell cycle is known as interphase, and interphase is when the cell is kind of just like doing its own thing, and it's doing what cells do, which varies depending on the cell you're talking about. But each cell has its own job, and then if that cell needs to make a copy of itself, either because you're growing or because the cell becomes damaged or something, then it enters into a different part of the cell cycle. And the part of the cell cycle where the actual DNA comes out of the nucleus and creates two separate cells is known as mitosis. The copying of the DNA actually happens earlier on, happens during a part of the cell cycle known as the S phase, which is part of interphase. So during that part, the DNA replicates, so you get two copies of DNA, and the cell gets bigger. And then the nucleus is going to split open, the DNA will divide, and two new cells will form in mitosis itself. It talks a bit about the different phases of the cell cycle here, or of the of mitosis here. You've got prophase, metaphase, anaphase, and telophase. Those are parts of mitosis. Prior to mitosis, remember, is interphase, which is made up of the G1, S, and G2 phases. And then after mitosis, when the cell splits apart into two new cells, we call that part cytokinesis. Prophase is just kind of getting ready. So first of all, when DNA is in interphase, it's made up of these long, kind of stringy strands of DNA, right? And in this part, it's easier to be read, to be transcribed and translated, right? Because protein synthesis is happening all the time because it's all stretched out. But what happens is when you start to make copies and then divide the cell up, those long stringy strands of DNA can more easily become entangled with each other, and you don't want that to happen. That's going to lead to mutations, and chunks of the DNA can get ripped off, and it's just like bad news, right? So the first part during prophase that happens is that DNA shortens and becomes condensed in what is known as chromosomes. So the chromosomes that you've heard about are literally just DNA that's been kind of coiled and wrapped up so that it's more easy to be divided, and you can take half of it and put it in one new cell and half and put it in another new cell, and it doesn't get all tangled up with each other, right? That's important. So that happens in prophase, and so we start to see chromosomes becoming visible under a microscope. The other thing that kind of happens, which is pretty cool, is that something called the mitotic spindle forms. It's made up of these things called microtubules. And I think of them, sorry, I'm using an object here as a cell just because I happen to have a giant bag of potatoes next to me, which isn't normal, but it happened for today. I think of this as almost like robot hands, right? So you got these spindle that forms on both sides of the cell, and these little microtubules, they work to kind of move around the chromosomes. They come in and they grab a hold of a chromosome and they can move them around in the cell. They can push them and line them up in the middle and then later pull them apart. And so they kind of act as little tools, as little robot hands. That's what forms during prophase. And during metaphase, those spindles take the chromosomes and line them up at the center of the cell. So metaphase, which is up here, means middle, and middle, they're lined up in the middle. And then we have, they start to pull apart to opposite ends of the cell, and that is anaphase. And eventually, the nuclear membrane is going to reform, and then the chromosomes start to unravel, right? So you no longer have those compacted chromosomes. You now have the stringy structures you had before. And this takes place in telophase. And then the whole kind of cell thing cleaves apart into two new cells, and that's called cytokinesis. And the whole process of mitosis takes just a couple hours or so. It's actually pretty quick, the actual division process that takes place. So the point of the division of mitosis is to make an exact copy, makes a clone of the original cell. And again, this happens for multiple different reasons, one of which is growth. Like when an organism is growing in utero, it starts off as a single blastocyst, or it starts off as a single zygote and eventually becomes a cluster of cells, a big ball of cells known as a blastocyst. Okay, now the thing is, when this happens, at this point, the cells are still pretty much all the same. So cells are kind of generic still. So they have to go through a process known as differentiation in order to become specialized at the job that they're going to be doing. So there's two terms I want you to be familiar with here, proliferation and differentiation, both pretty long words, right? So proliferating is when cells are being reproduced by mitotic division. They're growing and becoming numerous. So proliferation literally means growth or like multiplication of something. And then differentiation is when the cells actually become their specialized version. So different cells have different functions. Cells that are in the lining of your stomach, their function, there's multiple kinds of cells there, but maybe one of them has the function of producing stomach acid. The cells that are on my skin, I don't want them to produce stomach acid. That'd be bad, okay? Right? It'd be bad to do that. So they don't do that. So different cells have different functions. Let's see here. All right, so development, it says, begins in the womb or in the uterus, and all organisms begin as a single cell called a zygote, and through mitosis, the zygote divides until it becomes a blob of cells known as a blastocyst. And it says, how does this blastocyst become a zebra? The zebra zygote is what is known as a stem cell. You probably heard the term stem cell before. Stem cell, think of it as like being kind of like a generic cell that can become any other kind of cell. We have different categories of stem cells. There's totipotent, pluripotent, and multipotent stem cells. So different types of stem cells can have more or less genericness, if you will. Totipotent stem cells are the first stem cells to arise in the developing embryo. So these can differentiate into any other type of cell, and they can also become cells that are not part of the embryo itself. In this example, they become like the placenta. So the placenta is another organ that actually is outside of the fetus that gives the nutrients to the fetus and supports it while it's in utero. And then we have a close cousin, pluripotent cells, and pluripotent cells can develop into any other type of cell except those extra embryonic cells. So it can't become like the umbilical cord or the placenta or anything like that. It can only become any other type of cell in the body. They're still pretty moldable and pretty generic. And then you have multipotent stem cells. Multipotent stem cells are the stem cells that we have in our bodies right now. And they're already somewhat specialized. So they can become kind of just a handful of different kinds of cells. For example, you might have some multipotent skin cells that only can become, different types of cells within your skin. Or maybe a multipotent blood cell can become like a white blood cell or a red blood cell or something like that, but it can't become a skin cell. So it has just a certain number of cells it can turn into. It's still not just one type. There's multiple types, but it's a limited number. In adults or anything other than an embryo, there are no pluripotent cells left. And so pluripotent cells are kind of cool because they can become any other type of cell. So they can ideally change into something that we might need to repair that maybe we don't have the ability to do. So we'll talk a bit more about, in a different lesson, something called induced pluripotency or pluripotency. I don't know how you pronounce it. And basically what this is, it was a scientific breakthrough happened a decade or so ago, where they have the ability now to take adult stem cells, like from your skin, and then use certain enzymes to reverse time kind of and rewind them and turn them back into a pluripotent cell. So now instead of just becoming a couple of different kinds, they can become any other type. And they've been able to do this to produce like take one of your skin cells, for example, and produce like a little tiny micro heart. And the idea is that in the future, if they can figure out how to grow these and how to get the structure right and stuff like that, they should be able to take this process and let's say you needed a heart transplant to actually grow you a brand new heart that's exactly the same genetically as your heart. So it wouldn't be a donor organ and you wouldn't have to reject it and there wouldn't be any of that kind of stuff going on because it's your body cells reproducing. So that's the future of some of this genetic engineering. Let's see here. So just as a reminder that your chromosomes are made up of DNA, deoxyribonucleic acid. And as we talked about transcription and translation before, it's basically an instruction manual to make you, right? And every cell in your body contains the entire library of all the information needed to make your body. But you don't use that everywhere. As I gave that example of you don't express stomach acid on your face, right? So the process in which a cell differentiates by reading just a little part of the code is called gene expression. The code isn't read completely everywhere. It's only read in little chunks. Let's see here. Okay, so now it talks about how, mitosis is used in a growing organism, a developing organism to reach maturity, to reach adulthood. That's how an embryo grows into a fetus and how a baby grows up into an adult, right? But once you reach adulthood, you don't need mitosis to grow and develop anymore, but mitosis still plays an important role in your body. It is used in cell repair and replacement. In fact, you replace your cells, many of your cells in your body are replaced, and the lifetime of those cells varies. So, like skin cells can take 2 to 3 weeks to be replaced, but other cells can take anywhere from months to years to get replaced. Just it kind of depends on how long that cell lives. Skin, as you can imagine, it's exposed to the environment. It's in a really, really rough place, and so it has to get replaced more steadily and more readily. Cells that are more protected, that are inside your body, don't get the same length of time. They have a longer lifespan because they are in a more controlled environment. At some point, though, a cell will replicate, make copies of itself to replace itself when it starts to come to the end of its life cycle, and you'll have a brand new cell. But at some point, a cell reaches the end of when a copy from that cell can be made, and it won't have errors in it anymore. You don't want to make copies too many times in a row, or it starts to affect how good the copies actually are. And also, cells that become damaged, you don't want to make copies of those either, because again, you don't want more damaged cells, right? So when this time is reached, we call this state a senescent cell. So a senescent cell is one that can no longer reproduce itself and fulfill its function anymore. And at some point, that cell will just continue to live until it can't anymore, and it will die. Um, you don't want, again, to reproduce cells that are in bad shape. All right, so that gets us to the check-in, the practice, and I believe you can read a little bit here on skin grafting, which is kind of a cool technique, and that's pretty much it for this lesson. I think I've answered, yep, all the different parts for this lesson. If you've got questions, you guys know how to get in touch with me. Otherwise, have a great day, and we'll see you in the next one.